Hi students. Adaptive immunity players display four unique properties: specificity, diversity, differentiation of self from non-self, and memory. Both B and T lymphocytes primarily shoulder the responsibility of adaptive immunity absolutely amazingly. B cells have two major functions. They, as APCs, present antigens to T cells, and more importantly, they produce antibodies to neutralize infectious microbes. Antibodies coat the surface of a pathogen and serve three major roles: neutralization, opsonization, and complement activation. Neutralization occurs when antibodies cover the pathogen entirely, rendering it ineffective and unable to bind and infect host cells. In opsonization, an antibody-bound pathogen is located much more easily by phagocytotic immune cells such as neutrophils and macrophages, since they possess Fc receptors that bind to the free Fc portions of antibodies coating the particulate antigen. Likewise, the classical pathway of complement activation involves attachment of the first complement C1 to the Fc portion of the antibody bound to the antigen. This manifests in the formation of a membrane attack complex or MAC on the pathogen surface, thereby causing loss of cell integrity and finally death of the pathogen. Antibodies are expressed by B lymphocytes in two ways: the membrane-bound form or MIG, also termed B cell receptor, which sits on the surface of a B cell, and the other form is SIG or soluble immunoglobulin, which is generally termed the antibody. The purpose of switching of B cell appended MIG into free and soluble form SIG is to efficiently find the pathogens and clear them. This dual expression is very important, as first the BCR recognizes the invading antigen and binds to the same. This activates the B cell, eventually causing the switch from MIG to SIG. All antibodies are unique with respect to their antigen binding site, and each B cell can express 5 isotypes with identical ABS, that is IgG, A, M, D, and IgE. While they may have overlapping roles, IgG is generally important for neutralization, opsonization, and complement activation. IgA is found in body secretions and does neutralization in the gastrointestinal tract. IgM is the first antibody to be produced in the body and carries out complement activation. IgD is involved in activating basophils, and IgE is necessary for activating mast cells for release of pharmacologically important chemicals causing hypersensitivity type 1. Now guys, moving on to T cells, these have a variety of roles and are classified by subsets. T cells are divided into two broad categories: T helper or CD4+ and C cytotoxic or CD8+ cells based on their surface proteins. They carry out multiple functions, including stimulating other immune cells and killing infected cells, respectively. CD8+ T cells upon activation become aggressive and are termed cytotoxic lymphocytes or CTLs. These are crucial for recognizing and eliminating virus-infected cells and cancer cells via apoptosis or programmed cell death. The four major CD4 T cell subsets are Th1, Th2, Th17 and Treg, with Th referring to T helper. Th1 cells are critical for coordinating immune responses against intracellular pathogens by activating phagocytes and also the altered self-cell destruction by TC cells. Th2 cells interact with B cells and help them to divide, differentiate, and secrete antibodies. Th17 cells are important for recruiting neutrophils, and they are named so since they secrete interleukin 17 or IL-17. Lastly, regulatory T cells or Treg, as the name suggests, serve to modulate and, if required, even inhibit the activity of other T cells. They prevent adverse immune activation or overactivation and maintain tolerance or prevention of immune responses against body's own cells and antigens. Guys, hope this description was easy to understand. I thank Brahmjeet for appealing visuals. Together we'll be back soon with more. Till then, keep learning and growing. It's bye till next time.